I want to welcome Ryan Frankie tonight. Ryan is a longtime MMS member. He grew up in Eagan, Minnesota, and learned to revere the natural world at a young age. During a fall camping trip around Lake Superior in 2015, he became fascinated with fungi and just shortly thereafter joined the Minnesota Mycological Society. Since he joined, he has opened a gourmet mushroom cultivation business. He edited the MMS newsletter for us from 2018 to 2020, and most recently joined the mycology-focused Forest Pathology Lab at the U of M. Last year, he was one of MMS's graduate scholarship recipients, where he introduced us to his research, which addresses the key question: can fungi help us get rid of invasive buckthorn, non-native buckthorn? Tonight, he'll present findings from the past 2 years of research. So, Ryan, thank you so much for being here tonight. We're excited to hear what's new in your research.
Yeah, thanks for the introduction, Kathy. It's great to be back here presenting about a year later, more recent developments with our project to develop a mycoherbicide for common buckthorn. This is a project mainly between myself and my PI, Bob Blanchette, but also with many other collaborators, undergraduates, students in our lab, and citizen scientists, and city and county governments. And it's been a real joy to work on this project for my PhD here at the University of Minnesota. So first, Kathy gave kind of a comprehensive overview of my background, but I guess here it is again. So I became interested in trees at a very young age, just spending time outdoors. And I received my BA in anthropology and sociology from Gustavus Adolphus College in 2014. And I didn't really become interested in fungi until 2016 on that infamous camping trip around Lake Superior, when the mushrooms were so numerous and conspicuous, you'd have to be blind to not see them. It was a beautiful experience. And I shortly thereafter began cultivating mushrooms as a hobby, which turned into a business. And I did that for a few years. And I actually ran an all mushroom CSA for 50 households in 2020. However, I shuttered the business after that season of CSA because I was interested in doing research. And if you know any mushroom growers, it's a lot of work. And I wasn't finding much time to do research outside of production. And so I started with a bioinoculant company called Terramax as a lab tech in 2020. And Terramax produces mainly bacterial-based inoculants for conventional crops like corn and soybean to help them grow bigger and faster. And at that time, I also began going back to school at my local community college for biology, just taking some general bio courses. And after I left Terramax to come to the University of Minnesota as a researcher in an ecology lab, the Boris Bloom lab. And I did that for a year and picked up a bunch of skills in that lab, processing data and analyzing data. And at that time, as an employee, I was able to take a course here at the University at a very discounted rate. And I decided to take diseases of forest and shade trees. And that is with my current advisor, Bob Blanchette, who's been teaching that class for a long time and continues to teach it. And I just, further fell into a further fascination with trees and fungi, and specifically with fungi that cause disease in trees. And I got talking with Bob after class one day and realized we had a kind of a shared interest in biological control of invasive trees. And Bob had recently gotten a grant to look at developing a mycoherbicide for invasive buckthorn. and asked if I wanted to come onto the project as a graduate student. And I said, heck yeah. And so I applied to the program and got in that spring of 2023 and have been in the lab since, working on my PhD and developing a microherbicide, which has been a lot of fun. And you can see here's in this photo is myself and my undergraduate worker, Jenny, who's been on the project for about a year and a half now and has really been instrumental in getting this research done. So many thanks to Jenny. So I want to just kind of preface my research by saying that forest pathology as a discipline has historically focused on trying to understand fungi that cause calamities in our forests. And there are plenty of prime examples of this happening. For example, we have butternut canker caused by the fungus Ophiogonomia clavigenetai jugulandocerum, which emerged as a pathogen in the 1960s in North America. It still has, scientists still don't understand where it came from exactly. However, this is a devastating disease of butternut, and it causes cankers in the stem. And you can see in the photo on the right here, there's all of these basically areas where the fungus has colonized and killed the cambium tissue. And so the tree can no longer regenerate new tissue there, and it's not able to transport nutrients up and down the stem. And if there's enough of these cankers coalesce, basically it girdles the stem and kills the stem. And if you've ever seen a butternut without this disease, you're a lucky person. Butternuts have been vastly reduced in their abundance in their native range, and it's kind of a sad story because they produce these delicious things called butternuts, which I've eaten myself, and they're really delicious. They're buttery, kind of a mild, nutty flavor, and they're enjoyed by wildlife. Another example of a wily and destructive forest pathogen is the fungus that causes chestnut blight. This fungus is Cryphonectria parasitica, and this emerged as a pathogen in the North America in the 1920s and has really reduced what was once a keystone species and sentinel tree in primarily the Appalachian Mountains and surrounding areas to just a virtually functionally extinct species. And so you can see the photo on the left here are these huge chestnut trees. And this photo was taken in 1910 prior to the outbreak of chestnut blight. And the photo on the right is the current state of affairs for chestnut. And you can see these trees have not gone extinct in the area. They survive as young adolescent trees until they become infected with the fungus. And the fungus will grow in a diffuse canker, killing the cambium faster than the fungus can wall it off and kill the tree down to the root collar. And the fungus cannot penetrate down to the roots. And so the chestnut survives as these stump sprouts, which is also a very sad story. And currently there aren't really any scalable remedies for either of these diseases for our trees. So my research as a forest pathologist asks a different kind of question. Instead of trying to mitigate damage from forest pathogens to the trees that we love, we want to know, can we leverage the destructive power of forest pathogens to control unwanted, non-local plants in our forest? So meet the cast of characters here. We have common buckthorn on the left and glossy buckthorn in the middle here. And these are both non-local invasive species of buckthorn that are present here in Minnesota. We also have a third species of buckthorn here in Minnesota This is a native species. It's desirable. It's alder-leaved buckthorn. And in contrast to its non-local relatives, it is very short-statured. It only grows to about 3 feet tall. And it grows in a wetland habitat and it reproduces via rhizomes underground. And so they produce these large spreading clones in wetland habitat, as opposed to their non-local relatives, common and glossy buckthorn, that grow as singleton individuals into essentially into small trees or large shrubs. And so hopefully, yes, we can leverage the power of destructive pathogens to control these two unwanted species of buckthorn in our state. So this is a general outline for the rest of my presentation. Next, I'm going to introduce common buckthorn and it's the impacts that it has on the ecosystems here in Minnesota. And after we've become more thoroughly acquainted with common buckthorn, I will jump into our research to develop a mycoherbicide for common buckthorn. And this research is being conducted in 3 stages. First, we have collected a variety of fungi from dead and dying non-local buckthorn. And this stage is primarily in the past tense at this point. Most of our sampling is done. Although if you have some dying buckthorn that you know of, give me a shout and I'd be happy to come out and take a look. Maybe there's a fungus that we've missed so far in our survey. And so second, we're testing these fungi for their ability to cause disease on healthy buckthorn. So just because we've isolated a fungus from dying buckthorn does not mean that it is causing the decline of the buckthorn. Some fungi may be secondary pathogens or simply surviving on already dead tissue in a declined buckthorn. And so we want to know which of the fungi that we isolated in step 1 are highly virulent on buckthorn. And to do this, we basically inoculate these fungi into healthy buckthorn and measure the result. Third, we are developing, actually I say one methodology, but various methodologies for application of these fungi on buckthorn at field scales. And that is really, to me, kind of the most fun part of this research is taking our collection of fungi that we know to cause disease on buckthorn and exploring ways to make them useful and available for land managers to actually do the work of controlling buckthorn. And lastly, I will end with some future research directions. Common buckthorn was brought over to North America in the late 1700s by European immigrants. It was valued as a medicinal plant. The berries were considered a cathartic, hence the species epithet cathartica. And I had to look up what exactly is a cathartic, and it means something that makes you poop. And so essentially, Europeans were bringing this plant over and planting it in their gardens. One historical record shows a doctor had this in his yard and would basically prescribe these berries to his patients, and these berries were helping people poop. And you know, I guess it's the little things sometimes. And in this case, drove a big change because this plant quickly naturalized in the eastern seaboard of North America, where it was first brought over. And then it spread further west across North America, both in Canada and in the US, was facilitated by the movement of settlers. And settlers were actually valuing this plant not necessarily for its medicinal properties, but for its what should I say? Um, its usefulness as a good and hardy, so cold hardy and insect and pathogen resistant hedge plant. And it has since naturalized, in much of northeastern and upper Midwestern states, and in addition to various provinces in eastern and central Canada, including Minnesota. So this tree has become invasive in many different habitats in a lot of US Midwestern and Northeastern states. And you can see this is a map here showing states that have designated this species as a problem. And so in the red color, we have states, including Minnesota, that have prohibited the importation, sale, and transportation and propagation of this tree in the state. And then in in the salmon color, we have states that basically designate this as a problematic tree, but do not prohibit its propagation or sale or importation. And then states in black also prohibits its sale and importation and propagation, and additionally have it on a state list. And so, this, I think, picture speaks volumes about the problems that common buckthorn creates in the environments that it takes hold. Nicely put, common buckthorn has a competitive spirit. If you enjoy walking or hiking through our Minnesota parks, something akin to this image is likely familiar to you. Common buckthorn is a quick arrival to disturbed sites. Its fruits are readily dispersed by a multitude of bird species in late summer, fall, and winter. And once a few arrivals become established and begin producing fruits on a disturbed site, seedlings are quickly recruited to the site in mass and grow to form dense thickets, which is what you see here. Common buckthorn is broadly adaptable and can do quite well for itself in a variety of ecological conditions. Hence, it has established itself in a variety of habitats in its introduced range. Common buckthorn is drought tolerant, moderately flood tolerant, and extremely shade tolerant. And in fact, common buckthorn defies the typical physiological trade-off between shade tolerance and responsiveness to light. And what this means is that saplings can both persist under closed canopies and respond to gap formation in the canopy with a rapid growth, faster than our native tree species, shade-tolerant tree species like sugar maples and basswoods. And so as a result, we're basically having less biodiversity in our forests, but also less canopy tree seedling recruitment. So the canopy trees are not able to regenerate because the buckthorn's persisting in the understory and then outcompeting those native canopy trees when there's a gap formed in the canopy. So the interaction between these traits, namely dispersal, fecundity, growth, tolerance, and others with the landscape is associated, as I said, with decreased plant diversity in the forest understory, as well as depresses native canopy tree recruitment. And I want to say, too, with this picture here, this photo, you have some aspen trees, which are an early succession species in our forests. And you have a complete understory dominated by buckthorn. And normally these aspen trees are succeeded by trees such as maples and basswoods, ash, oaks. However, in this case, the seedlings that would be saplings of those native trees really don't have a chance. And so essentially natural succession in the forest is arrested by the colonization of common buckthorn. So there are a few, I should say, many different methods that are currently used to control common buckthorn. And I'll say a little bit about the ones on the screen here. We have mechanical removal, which is effective because you pull the entire tree out of the ground. It doesn't have any roots. It doesn't have, it's not able to, stay hydrated and transpirate and photosynthesize, so it dies. However, this is, incredibly intensive, labor intensive, and time intensive, and is really not feasible to accomplish at a large scale. We also have prescribed burning, which unfortunately, it does work for smaller, very small saplings and seedlings, but once the saplings get more than a quarter inch in girth, the burns, low-level burns, actually don't kill the root collar, and those saplings just sprout back. And so it's not a super effective technique. Goat browsing has actually been found by researchers here at the University of Minnesota, led by Daniel Larkin, to increase the density of common buckthorn where goats have browsed. Unfortunately, because it's kind of a cute and endearing tactic, but the research shows that it's ineffective. And so the goats, they're not able to kill the root collar. So the goats come through and they eat the leaves off and strip the bark and might kill the plant down to the soil level, but the plant has carbohydrate reserves in its roots and is able to resprout a new flush of leaves. And it would really take an careful, planned timing of successive goat browses to be able to kill a buckthorn plant. And unfortunately, I haven't, I've seen a lot of places that have been goat browsed, and they are terrible buckthorn thickets. And so doesn't appear to me, and preliminary research shows that it's really not a great method to control common buckthorn. Now, chemical herbicides, while they are an effective and common tactic, like glyphosate or triclopyr-3, they are not always appropriate for sensitive sites where they can contaminate the environment or harm non-target vegetation. And so that leads us to kind of, the problem that we're addressing here is developing an effective and safe mycoherbicide for buckthorn. And so I'll first talk about our efforts to collect fungi from dead and dying buckthorn. And so our approach to developing a mycoherbicide for buckthorn rests on this hypothesis that when an invasive species comes to its new range, it quickly expands across that range because it has some advantageous life history traits. Also, perhaps because it's left some of its natural enemies behind in its home range. So insects and pathogens that infect it in its home range are not in its introduced range. And so it has some kind of competitive abilities. However, there's part of this hypothesis about invasive species, the enemy release hypothesis, is that even though it may be released from its natural enemies in its home range, it also accumulates new natural enemies in its introduced range as it spreads across space and comes into contact with new potential pathogens, and it is likely to accumulate new pathogens, natural enemies. And so buckthorn has been on the continent for 225 years, and we thought, it's worth a shot collecting these fungi that are associated with declining buckthorn and attempting to develop a mycoherbicide from these native, naturally occurring fungi. We had the kind of inception of this project is back when the DNR and the MDA contacted my PI and said, "Hey, we have some sites where buckthorn is declining, and maybe this is useful information. And so basically that small idea has grown into this large project that, as you will see later in this presentation, has some very promising preliminary results. So let's talk about this, this collection of fungi. We derived our collection from 19 sites that we sampled across the states of Minnesota and Wisconsin, which are marked there in blue on the map. 15 of these sites we sampled, there was exclusively common buckthorn growing that was declining. And at 3 sites, there was glossy buckthorn that we sampled. And at 1 site, we sampled both species. There should be another bullet there, but there's an additional site with native buckthorn that we sampled, and that is at a site in Wisconsin. We had many collaborators in locating these sites and helping us doing sampling, including the Minnesota Department of Natural Resources, the Minnesota Department of Ag, the ecological restoration nonprofit Friends of the Mississippi River, various citizen scientists who just called us up and said, hey, we got some dying buckthorn in the backyard, and we were invited out and sampled in some of those backyards. And also the Wisconsin Department of Natural Resource was a key collaborator for our sites in Wisconsin. This is a nice photo collage of some of the symptoms that we saw while we were sampling. On the left, you see I'm standing with quite a decent sized buckthorn tree, at least 5 or 6 or 7 years old, and it is completely died. And it's not getting a lot of shade there. Something is killed that tree. And in the middle, we have a section of stem that has an elongate canker, and you can see basically in the middle of the canker, there's all this basically dead tissue where the fungus has killed the cambium. And then on either side, the tree has formed this callus tissue, which is essentially like a chemically and physically strengthened tissue that the plant forms to wall off the fungal infection. Over on the top right, we have a cut face of a stem where there's a canker that has killed approximately half the circumference of that stem. You can see kind of where it's all black here, and the lighter color is the healthy sapwood and cambium tissue. And then the photo beneath that depicts a buckthorn stem that has been cut that has multiple canker infections that had occurred over multiple years. So we used hand saws and pickaxes to take samples of these declining buckthorn. And you can see there's myself there with the pickaxe and my PI, Bob Blanchett, with the handsaw, pointing out a canker to Laura Van Riper, who is with the Minnesota DNR. And so we took these samples back to the Blanchett lab, where we isolated from symptomatic tissue. So you can see the photo on the very left is a glossy buckthorn stem, and there's a perennial canker there. And so you can see this kind of looks like a target. And basically what happens is the fungus has killed some tissue in the very middle here when it first infected this tree, and then the tree responded by forming that callus tissue around the initial infection. And it formed that tissue during the height of summer and photosynthesis, when the tree has an abundance of resource and is able to produce some of those secondary metabolites and produce a good defense to be able to produce that callus tissue. But then in the shoulder seasons, in fall and in spring, the fungus can keep growing at those cooler temperatures, but the plant doesn't grow at the same rate. And so it has a harder time protecting itself, and the fungus is able to overcome that defense of that callus tissue, and it kills that callus tissue. And the same thing happens the next year. During the growing season, the plant walls off the infection with callus tissue, and then the fungus kills it, and so on and so on. So you get these target-shaped cankers. Now, the photo in the center depicts a diffuse canker running lengthwise along a common buckthorn stem. And you can see the healthy phloem tissue of common buckthorn is this brilliant orange color. And it's been killed over on the left side of the stem, and so it's turned brown. And there's no clear callus tissue forming around that site of infection. And so what that potentially means is that the fungus is killing tissue faster than the tree can wall it off at all. And we've referred to this as a diffuse canker, which we saw a lot of this. A lot of these, at least a lot of our common buckthorn samples had this kind of symptomatic tissue of diffuse canker. And then the photo on the right is a common buckthorn root that has been cut in half and showing root rot. You can see it has kind of a mottled, punky appearance, and that's the wood being rotted. And so we isolated from some root rot too. Now, these graphs, or I should say bar charts, show the symptoms that we observed and that we observed on collected buckthorn samples. And so it's broken down by the buckthorn host. We have common buckthorn on the left. We collected 67 samples of common buckthorn. Glossy buckthorn on the right with 15 samples, and the native alderleaf buckthorn on the bottom with just 4 samples. I should say that these figures, as well as figures in successive slides, have been published in the open access journal titled Forests. And the publication is called Fungi Associated with Dying Buckthorn in North America. And because this is an open access journal, anyone can go on and type in the name of this publication and open it up and read the PDF for free, which is really great. And we're happy that we were able to do that. So coming back to these figures, so I should say, if you want to learn more about the details of this publication, go online and do that, because I'm just going to hit some of the key points. Coming back to these figures, for common buckthorn, you can see that primarily the symptoms that we saw on affected declining stems were diffuse cankers, with nearly 50 samples having a diffuse canker. Less so, I should say, the second most common symptom was a diffuse canker accompanied by fruiting bodies. Oftentimes, this was Urpex lacteus, the milky white tooth fungus. But also Peniophora genus was also forming fruiting bodies on some of our samples. We also had a couple of samples with root rot, including Xylaria polymorpha, dead man's fingers. And we had a couple of samples with diffuse canker and white rot of the sapwood, and a couple of samples with root rot and fruiting bodies. And that was that Xylaria polymorpha, the dead man's fingers. For glossy buckthorn, we have kind of a different story here. We had a lot of these localized perennial target cankers. That was the foremost symptom that we saw on collected stems, followed by diffuse cankers and fruiting bodies, followed by just diffuse canker, followed by root rot and localized canker with white rot. Now, for alderleaf buckthorn, we only were able to collect 4 samples because it is a native species, and it also doesn't grow close to the Twin Cities. You have to go out a ways to find it. And so we actually collected 4 healthy plants and 1 with a diffuse canker. And so we plated symptomatic tissue onto 3 different media types: SDA, BSA, and MEA+. And these 3 different media types select for different groups of fungi, because some groups, like ascomycetes, are much faster growing than basidiomycetes generally, and will overgrow basidiomycetes that may be coexisting in that symptomatic tissue. And so we use these selective medias to be able to discover slower growing media, or sorry, slower growing fungi that are present in our samples. And after we had the fungi grow out from the diseased section of wood that we placed down into the agar media, we basically subcultured a little piece of hyphae, hyphenated agar, into pure culture. And once we had pure cultures of our isolates, we extracted the DNA and amplified the internal transcribed spacer region of DNA, and then sequenced this region of DNA using the Sanger sequencer. And so I'll say a little bit about the ITS region of DNA is highly conserved in actually all eukaryotes. And in fungi, there's enough, I should say, difference in that region that it's been found to be very useful in separating species from one another. So there's enough similarity that you can compare them, but enough difference that you can tell one species from another. Additionally, we used an additional region of DNA, RPB2, to identify our Fusarium isolates, because the ITS region for Fusarium is just too similar between species to be able to make a species identification. So in total, we isolated 414 different individual fungi from our buckthorn samples. And you can see just from the photos here of these fungi growing in agar culture that there is a considerable diversity of fungi associated with declining buckthorn. And so far, we've identified, I don't know why this says 294, but so I apologize, this is incorrect. It's actually all 414 isolates have been identified as 89 unique taxa belonging to 64 genera. So we've finished identifying all of these isolates, and it turns out there's 89 unique species. And these 89 unique species belong to 64 genera. And I know there's a lot of seasoned mycologists in the audience tonight, but in case you're not, I just wanted to run through briefly how to conceptualize genus species, because a lot of my data and kind of revolves around this concept. And so a great example for this, because we all know morels, is the difference between a black morel and a yellow morel. And so both are in the genus Morchella, but the black morel has the species epithet of elata, basically designating it as a different species from the yellow morel, the Morchella esculenta. And so although these are the same genus and have a high degree of genetic similarity, they're not similar enough to be able to produce offspring. And you can see, the genus species kind of nested within taxonomical ranks, which you probably recall from high school biology. We have family above genus and order above family, class, phylum, kingdom, and domain. And so this is a nice illustration of all of our 89 genera visualized, I should say, compared to one another. I should say this shows the abundance of each genera. And so the larger boxes on the left represent genera for which we have more than that we collected. I should say we collected these genera from more than 17 samples. And so we have Clonostachys, Fusarium, which is a known pathogen of many plants, Alternaria, which is more so like a leaf pathogen or a saprophyte, Diaporthe, which is a canker pathogen of different woody plants, Nothophoma, another woody pathogen, and Phaeoacremonium. And so we have 17 individual fungi collected from these genera. And then the boxes kind of on the other side of the spectrum, these small boxes, these little squares, represent genera for which we collected one individual isolate each. And there's 35 of those genera. And then the boxes in between represent genera that we collected in more moderate abundances. And so you can see kind of from this illustration that there's a considerable diversity of fungi associated with declining buckthorn. And so which of these fungi may be useful to our project? Well, it's those that have been basically documented in the scientific literature as causing serious disease in woody hosts. And so this graph here is a breakdown of all of the genera in our collection by their lifestyle strategy. And so you can see that we collected 22 genera that can be ascribed to a canker pathogenic lifestyle, as far as we can tell by published literature. And this is the most abundant lifestyle in our collection. And this is followed by wood decay fungi with 13 genera. And you can think of this makes sense because as a pathogen kills cambium tissue, it exposes the wood beneath, and then wood decay fungi have a spot where they can enter and begin degrading the wood. And so it makes sense that we were also isolating wood decay fungi from diseased tissue. We also isolated a fair number of foliar pathogens, some endophytic fungi, which are known to exist within the tissues of plants but don't cause outward symptoms of disease. We isolated a couple of genera that can be characterized as living an entomopathogenic lifestyle, which means they kill insects. We also isolated a couple of mycoparasitic genera, which means fungi that parasitize other fungi. And we have a couple of genera of root pathogens, including Xylaria polymorpha, dead man's fingers, and Mycoleptodiscus terrestris, as well as Ramularia robusta. And we had a couple of soil saprophytes sneak in there. And then there's 23, or I should say 22 genera for which there's just not enough information available in the scientific literature to be able to place them into one of these neat bins. So the fungi that we're interested in are those that we considered either as a root rot pathogen or as a canker pathogen. And this Venn diagram encompasses all of those species. And so you can see in the kind of salmon colored circle, we have fungi that were collected from common buckthorn. In the blue, we have fungi collected from glossy buckthorn. And in the green, we have fungi collected from alder-leaved buckthorn. And in the overlapping spaces, we have those that were collected from multiple host species. And so you can see we have a considerable kind of bench of fungi for testing for potential to cause serious disease in healthy common buckthorn. And I should say that the numbers in parentheses next to each species name represents the abundance of that species. And if you would like to, study this a little bit more, go ahead and go online and search for that publication. And this figure is in there, so you can check it out. So to contextualize my study, it's helpful to know what is already known about, prior to my study, what is already known about fungi that are associated with common buckthorn. And really there is a great source for this. Really the only kind of comprehensive source for this is a PhD dissertation written by a student at the University of Western Ontario. And this student basically in their dissertation compiled, did a literature search and compiled all of the fungi known to associate with buckthorn in North America and in Europe. So remember, North America is the introduced invasive range, and Europe is the native range. And what she found was that there have been 22 species of fungi reported in the literature as growing on common buckthorn in North America, 33 species of fungi growing on common buckthorn in Europe, and just 2 species found growing on common buckthorn on both continents. You can see, I should say, moving down to the next bullet point, in her study, she basically walked around a few different woodlots in southern Ontario and collected fruiting bodies found on common buckthorn throughout the growing season. And you can see there's a nice photo collage of those fruiting bodies growing on common buckthorn. And ultimately, she found an additional 23 species known to associate with buckthorn in North America. Now, she mentioned the value of potentially exploring these fungi for their ability to act as a biocontrol, but she didn't actually culture these fungi and keep a collection. She identified them with via microscopic and macroscopic characteristics and just left it at that. This is a nice comparison of the fungi associated with common buckthorn that I collected in my study with those that have previously been reported on buckthorn. And so you can see on the y-axis we have, so this is, I should say this is genera. And on the y-axis we have the lifestyle for the genus. And on the x-axis we have the number of genera that have been collected on common buckthorn that can be ascribed to a given lifestyle. And the yellow bars represent the fungal genera that I collected in my study, and the blue bars represent the fungal genera associated with common buckthorn as recorded in the scientific literature previously. So kind of in summation here, my current study adds an additional 61 genera of fungi known to associate with common buckthorn, including 11 putatively canker pathogenic and 3 putatively root pathogenic fungi. And additionally, 11 canker pathogenic genera and 3 root pathogenic genera. And and these genera are comprised of 31 species known to cause serious disease on woody hosts. And so these are the fungi that we're interested in for their potential to act as biocontrols. And I have a few examples down here. For example, the canker pathogens collected in my study that are new to buckthorn include species in the genus Diporthe, Dothrella, Hymenocate, Urpex, Peniophora, and others. Now, we didn't just collect fungi from buckthorn. We broadened the scope of our collection to include fungal pathogens of non-buckthorn hosts. And we did this because there are some generalist pathogens out there that are wound ruderal pathogens that we didn't collect from buckthorn, but we thought still might be useful as a potential mycoherbicide. And so all of these native, naturally occurring pathogens were collected in Minnesota and in Wisconsin. And these include a few species in the Hymenocataceae family. And these species are known to cause a canker rot in their hosts, which basically means they both canker the stem, the cambium, and also rot the wood underneath the canker tissue. Also, Serena unicolor is another canker rot. Chondrosterium purpureum, another canker rot, and Verticillium dahliae, which is a wilt-causing fungus. Okay, so now that we've reviewed the fungi in our collection from dead and dying buckthorn and non-buckthorn hosts, I'm going to delve into a few of the studies that we've designed to test the ability of these fungi to cause disease in healthy buckthorn. So this is a big experiment we did last winter, so winter of 2023 and 2024, where we inoculated 246 common buckthorn trees. And I should mention that, at this point, everything in the presentation is new. So last year, I mostly presented on our collection of fungi from common buckthorn. And this year, I get to add a bunch of additional information regarding pathogenicity testing we've been using to assess the utility of these fungi to act as a mycoherbicide. And so 2 winters ago, we inoculated 246 common buckthorn trees with 31 fungi from dead and dying non-local buckthorn, glossy and common buckthorn, as well as 9 fungi from native buckthorn and 6 fungi from non-buckthorn hosts. We grow out these fungi on wood chips, and once these wood chips were myceliated, we made a small wound into the stem of the buckthorn sapling and inserted that myceliated wood chip, wrapped it with parafilm, which is like a wax paper to prevent the inoculum from desiccating, and then also wrapped the inoculation site with duct tape to protect from UV radiation. We harvested the saplings at 3 months post-inoculation. That gave time for the fungus to colonize and kill stem tissue. And then we measured the resulting size of the canker around that inoculation site as a length times width measurement. And what we found were really considerable sized cankers that were produced on these buckthorn saplings by a whole handful of fungi in our collection. And so this is kind of just to orient you to this chart here. On the bottom, we have the isolation source. So this is where this fungus was originally isolated from. And so we have, common buckthorn, glossy buckthorn, native buckthorn, and non-buckthorn hosts, namely maple and common buckthorn. And then above that line, we have the fungus itself, or the control. And so the first column there is the control treatment, which was just a sterile wood chip that we placed into the stem as a negative control. And then we have the picture, obviously. And then above the fungal pictures, we have the p-value associated with the size of the average size of the canker for that fungus compared to the negative control. And all of these treatments are significant. And so we can say without a doubt that yes, these fungi do cause disease in stem tissue when inoculated into common buckthorn. All of these the fungi are arranged in descending order of canker size. And so Fusarium stambicyanum formed the largest cankers, followed by Posicula frangulae, subspecies frangulae, which was isolated from glossy buckthorn, followed by Fusarium sporotrichoides, which was isolated from native buckthorn, followed by Serena unicolor, which was isolated from maple tree, followed by Urpex lacteus, which was isolated from common buckthorn. Now I have a bunch of more fungal names at the bottom of the slide because we conducted a very similar study this past winter with some additional fungal isolates that weren't yet in our collection at the time of the first greenhouse study. And these are the names of the fungi that produced significant size cankers in this second greenhouse study. So there's an additional 10 fungi here. So in total, we have 15 fungi that cause significant cankers when inoculated into common buckthorn. And these are our potential mycoherbicide candidate species. I was going to say something about one of these species, Mycoleptodistis terrestris, but I think for the sake of time, I'm going to skip over this slide. If I have time, I could come back to it. It's just kind of some fun information regarding the history of the species as a plant pathogen on other plants besides buckthorn. Now, I do want to talk about this fungus, Chondrotherium purpureum, because although this fungus did not produce significant sized cankers, which you can see in the photo on the right here, the canker is kind of this um, inner zone of dead tissue where the fungus killed the cambium. It did produce extensive staining in the sapwood. And so the cambium was healthy over the sapwood, but the sapwood was diseased. And we were consistently isolated the fungus from the staining in the sapwood. So the fungus is there, it's causing disease, it's moving through the vascular tissue of the tree. This fungal isolate was originally isolated from John Lampert's, or I should say John Lampert's son apple tree in Lakeville, which is a fun fact for all of us MMS members. I was very excited to get an isolate of this fungus and to find that John's son had one in his tree. And so I digress there. But basically, the reason why I want to point out this fungus and why we're interested in this fungus is because this is a pretty well-known and researched fungus as a pathogen of various hardwood trees. And there's this paper that was published in the 80s by Spires and Hopcroft that basically inoculated cut willow stems with the fungus, and the fungus grew down into the stem. And then at various time points after inoculation, the scientists took samples of the stem tissue and took micrographs using scanning electron microscopy. And you can see what they found was that the fungus actually grows and throughout the xylem vessels of the tree. And so this first photo was taken at 12 hours or 0.5 days post-inoculation. The second photo was taken at 28 days post-inoculation. And here you can see the fungus has ramified and formed kind of a knot of mycelium in the middle of that vessel. And this constricts water flow up to the leaves. And once the stem was killed, basically you can see this really thick conglomerate of hyphae in the vessel that is effectively constrained water flow to near zero in that stem, and the leaves wilt and the stem dies. And only at that point, after stem death, does the fungus grow out and colonize the cambium tissue. So canker size is not really a great metric for assessing the pathogenicity of this fungus. So as I said, Chondrotherium purpureum has been extensively researched. It is a global pathogen with a broad host range. And I want to point out that the photo here is of the fruiting bodies of this fungus, photographed by John Lampert on the crabapple tree in his son's front yard in Lakeville. This is the isolate, one of the isolates I've been working with. Another fun fact is that I actually found Chondrotherium purpureum killing the birch tree in my front yard. Not the whole tree, but a few branches. And so I was able to get another isolate of this fungus from the tree in my front yard, which is kind of bizarre that it would show up in my front yard because you don't see this fungus everywhere. But it's great to have a couple of isolates in our collection. So this fungus, has been explored as a mycoherbicide for unwanted hardwoods. And most recently, it's been explored as a mycoherbicide for common and glossy buckthorn. Now, this paper was published by a research group at Michigan Tech, and it was published in 2024, and is titled Assessing the Utility of a Native Pathogenic Fungus as a Biocontrol Alternative to Herbicide on Invasive Buckthorns in Forests of Upper Michigan. So great title, really descriptive. And basically what they did in their study design was that they cut buckthorn to stumps and then inoculated, or I should say applied a chemical herbicide to the stump, or applied a Chondrotherium purpureum agar slurry. And they actually did this not only to cut stumps, but also to girdle trees. And they applied the herbicide or the agar slurry to the wounds on the stump or on the girdle. And what they found was that the probability of mortality for glyphosate cut stumps was 94%. For glyphosate girdled trees was 81%. And unfortunately, Chondrosterum purpureum, when applied to a cut stump, resulted in 51% rate of mortality, which was significantly less than the glyphosate treatment. And the Chondrosterum purpureum, when applied to a girdled wound, resulted in 67% probability of mortality. And, frankly, it's not for a mycoherbicide to be adopted by the general public, by people who don't love fungi. The efficacy of the mycoherbicide needs to match that of a chemical herbicide. So these results may be slightly despairing. However, I want to point out that they only tried one inoculum formulation, namely an agar slurry. And so I think there are other ways to formulate this fungus that might allow it to be more effective when applied to buckthorn, which is something that we are exploring. Something else interesting in their study that they pulled out of their data was a significant negative relationship between percent canopy and the Chondrosterum purpureum cut stump treatment. And what I mean by that, and what this figure here is showing, is that for stumps that were treated with Chondrosterum purpureum, there were less sprouts that came out of that stump with a more shaded canopy. And so you can see the percent canopy on the x-axis, and as you approach 80% shaded canopy, you have, only, 2 or 3 stump sprouts. Or actually, I think this is 0 here. So you have almost complete depth of the tree versus in a more lighted environment with a thinner canopy, you have more stump sprouts, which kind of makes sense because the agar slurry, potentially, how the authors explain this is that maybe the agar slurry is drying out on the stump. And the sunlight is just harsh on the inoculum. And I think they're probably onto something in that conclusion. So, having read this research, I designed an experiment that controlled for the effects of environmental variables on the fungal inoculum, such as evaporative pressure and UV light. And in this controlled environment, I could ask the question, which, if any of our fungi, can colonize and necrotize the buckthorn root collar? As you know, buckthorn is a prolific resprouter when cut, and so an effective mycoherbicide needs to infiltrate the root collar and prevent resprouting. And so what I did was I grew out our fungi, 32 different fungi on sterile paper towels. And once those paper towels were myceliated, I cut buckthorn to stumps using a chainsaw, and then placed a piece of myceliated paper towel over the cut face of the stump, and wrapped the inoculation site with parafilm, and placed a nice reflective piece of duct tape over the top of the whole shebang. And so in all, I did this for 32 of our fungi, including top performing fungi from the greenhouse experiment, Fusarium samoscinum, Chondrosterum purpureum, and Herpex lacteus. I also included a negative control, which is just a sterile paper towel without any fungus growing on it, a positive control, Trichopyr 3, and in all, we inoculated 610 common buckthorn stumps using this method. The metrics that we will be collecting from the study are the number of stump sprouts per stump, as well as apparent mortality of the stump. And we implemented this experiment last July of 2024. And we're actually going to be collecting this data starting tomorrow and throughout the rest of the week. And so that's very exciting. Kind of a cliffhanger. I hate to leave you there, but we don't have the data yet. But I should say a preliminary look at the stumps that I made this spring showed a couple of fungal treatments that were killing 60% of the stumps or even up to 90 to 100% of the stumps. So I think we have fungi that um, theoretically in a sheltered environment can work as a mycoherbicide. I should say potentially can work as a mycoherbicide. So lastly, I know we're kind of short on time here. I want time for questions, but I'll just briefly uh, talk about some of the experiments that we're using to explore the possibility or the potential of these fungi to be applied at larger scales in the field. So one tool that we're using is the hypohatchet. This hatchet essentially makes a wound in a tree and squirts fungal inoculum all into that wound, all in one swoop. And for this experiment, we used spore and hyphal suspensions of 3 of our fungi. We inoculated 20 trees for fungal treatment, sterile water as a negative control, triclopyr-3 as a positive control, and then again, the metrics will be apparent mortality and number of stump sprouts. We'll be collecting that data in a couple of weeks. This is a video showing the action of this hypohatchet. So you can see, I want to point out just the hole in the hatchet there where the inoculum comes out. So you can watch it squirt out of there. It's kind of satisfying. Let's play it again there in case you missed it the first time. Yeah, so it's a neat tool. Oh, let's see. Okay, so for the sake of time, I guess I'll just quickly run through this slide. So when we set up this experiment, there was a big rainstorm that came through, and I was concerned that the rain washed the inoculum out of the wounds. And so what I did was I basically set up a second experiment with just one fungus, Fusarium sporotrichoides, and inoculated. I knew it was going to rain on like a Thursday afternoon. It was a high chance for a thunderstorm. And so I inoculated this fungus in the a couple hours before that storm hit. And then I did a second inoculation the following morning after the storm. And we got 2 inches of rain in that storm. And then I harvested these trees 5 weeks post inoculation. And I basically measured the rate of infection for each treatment group and the canker size. And you can see there was basically no difference in the rate of infection of trees that were inoculated before the rainstorm versus after the rainstorm. So the spores of this fungus were able to kind of get into those vessels and were sheltered from the rain, which is kind of neat. And you can see here, there's a photograph of just after 5 weeks of growth, this fungus has really grown out from that initial wound site and is cankering and killing the cambium. And you can see in the photo on the right, the sapwood here is stained pink. And the fungus was consistently isolated from that sapwood. So this fungus, similar to Chondrosterium purpureum, is growing throughout the vessels of the wood, which is great. And is causing disease there. Okay, so briefly to touch on our future research directions for this summer. We are, like I mentioned, recording the results of the cut stump and hypohatchet experiments that we set up last summer. We are also testing various inoculum formulations of our top performing fungi on cut stumps. And in a similar vein, we are testing various inoculum formulations of top performing fungi using the hypohatchet. As I kind of mentioned, and one of the initial slides of my presentation, there's been just a lot of people and organizations that have been that have made very significant contributions to this research in a myriad of ways. So from locating sites with declining buckthorn to allowing us to set up field experiments on their properties, to the undergraduates in our lab, putting in lots of hours at the lab bench or out with me in the field. My fellow graduate students, just for their camaraderie and advice. Robert Blanchett, of course, my PI for his um, steadfast support and deep well of ideas. And Ben Held, our research scientist in the lab, he always makes sure that we have the things that we need ordered and in stock, and is it's also a deep well of good advice. And lastly, my mom and dad for getting me interested. I should say, getting me outside a lot when I was young. And I think um, because of their encouragement to spend time outside, I developed a deep affinity for trees. And that is kind of what has led me into this research. And last but not least, my wife, Alicia, for her moral support, and our 2-year-old son, Malachi. Actually, he's 3 years old now. This is a slide from a different presentation. And a little bit more spunky at 3. And he's just both the most inspiring and also humbling force in my life. So with that, okay, so I want to acknowledge, our funding for this project comes from the Minnesota Invasive Terrestrial Plants and Pest Center, which is in turn funded through the Minnesota Environment and Natural Resources Trust Fund, which gets its money from the Minnesota State Lottery. So yay. And yeah, we'll take some questions. I can stay on. I know I went a little bit over. I could stay on till 8:40. So we got 15 minutes for questions for those of you that want to stay a little bit later and have some curiosities about the research.
Awesome. Thanks, Ryan. Oh, great. Yes. If you stop sharing, I will maybe just, there's a couple questions in the chat, and I would invite those people. Howard, you had a question. I think yours was the first one in there. How widely are the, yep. Do you want to just unmute and ask?
I did. I was just curious. These seem to be fairly universal fungi in terms of their hosts, and I'm wondering, how do you separate one from the other than finding an inoculation process, which you have been doing, so we're not harming other beneficial or native tree species or woody species.
Yeah, so I believe the question, Howard, that you're asking is like, how do we apply these pathogenic fungi in the environment in a way that doesn't endanger non-target plant species? And I have to kind of take a nod to a Canadian researcher, Becker, who published in 2005 a paper regarding an experiment that she conducted with Chondrosteria purpureum. And in this experiment, she cut Chondrosteria purpureum to stumps, or I should say, sorry, cut speckled alder to stumps and inoculated with Chondrosteria purpureum, and allowed the Chondrosteria purpureum to colonize and kill those stumps with like a 90% mortality rate over the course of a year. And then the following year, during the fall, the Chondrosteria purpureum started fruiting on those stumps. And at that time, she went around the perimeter at various distances. She, I think, cut 3 perimeters at various distances from that experimental plot. And she cut speckled alder to stumps. And these were basically to act as like trap trees for those spores to see if they would be infected, which is kind of a fun idea. And what she found was that some of these trees did become infected with Chondrosteria purpureum, but actually it's because this fungus is a natively occurring fungus, there's a lot of locally adapted spores already present in the area. And when she did the genetic analysis of the fungi that infected those trap trees, those fungi were actually not the isolate that she used in her experiment. So essentially, she concluded that for the fungus that she used, her isolate had no impact on the surrounding vegetation, even when fresh wounds were made, at the time of fruiting. So I don't know, does
That
that
could,
answer your question?
yeah, that answers it. It could be that there's a genetic trait specific to the buckthorn, or it's an adaptation that may not make it uh, as likely to infect it as a native purpurium might. I don't know.
Yeah,
Interesting.
yeah.
Yeah. Howard, your question made me think too that it was like, it'd be interesting to know if there are any fungi that were brought over with the buckthorn that weren't already here, which would be a harder
Oh,
history thing, but it's, anyway.
Absolutely. Yeah, that's
Yeah.
definitely an interesting question.
So the next question is from Mark, and he says, is the buckthorn bark sanitized with bleach or another chemical prior to the inoculation? And Mark, you're welcome to unmute as well if you want to add anything to that question.
It's actually Carol. I'm on Mark's computer.
Oh, hi. Hi, Carolyn.
Hi. Yeah, I'm just wondering if there's anything done to prevent cross-contamination, like washing with bleach, or is that not really something that you worry about with fungi?
Yeah, I guess so it's not a common practice, I guess, to do that in inoculation experiments that assess the pathogenicity of a fungus on a tree. And I think like basically, I guess the reasons why you would want to sanitize or why you potentially would want to sanitize would be to kill any fungal inoculum from a different treatment that you're inoculating that same day. But I haven't had a problem with that. I haven't like pulled chondrosterium out of a stem that I inoculated with fusarium. So that hasn't been a problem. I guess another reason why you potentially would want to do that is you'd want to kill any like mycoparasitic fungi that might be existing on the stem, like trichoderma. But I think that in practice, at a field scale, it doesn't make sense to go around sanitizing trees before you would inoculate them with a microherbicide. I think it'd just be kind of an extra step, and it would be harder to get people to adopt it at a larger scale. And so, yeah, I think, and if the fungus is also to that consideration, these fungi are plant pathogens and they're able to get into the plant tissue where other fungi like mycoparasites can't access because they would succumb to plant defenses. So I don't think that's a problem.
Okay, thank you.
And then Aaron's question, how many years do you think it would be before we could apply a mycoherbicide to kill buckthorn at a large scale?
Oh man. I don't know. I mean, it's just the research is still underway, and we don't. I guess if I were to take a wild guess, I don't know, 5 to 10 years or something like that. I mean, there's even if we had a really promising fungus and a promising methodology that was efficient and efficacious, if it were commercialized, the company to commercialize it would have to register the microherbicide with the EPA. Which is a audacious undertaking. I've read some of those registrations, and there's just a lot of information that has to be provided, which is a good thing because it keeps our environment safe. So yeah, it's hard to say.
So once you do get to the place, there's this whole other step and this whole other process that has to happen to launch it.
Exactly.
Fascinating. Awesome. And Julie's asking, if we find small buckthorn seedlings while foraging on state land, should we pull them?
Buckthorn seedlings on state land? Hell yeah. Yeah, get them out of there. Yeah.
Yeah, it is amazing because I do feel like, I don't know if other people have this experience, John and Claudette, I think we've talked about this. When I go to places where there wasn't buckthorn and I was like, say, picking chanterelles or other mycorrhizal species, and then once buckthorn kind of starts to fill in, I don't really find the mycorrhizal species in the same way. I feel like it must interrupt. One, you can't really walk and forage in the same way. It's just not pleasant. But two, I wonder if it kind of interrupts, the root growth of the mushrooms.
I'd have to say that's what we have observed as well.
Mhm.
Yeah, me too. I feel like
Yeah.
it
Now,
gets,
this is Howard. I found the chanterelles prolific in the midst of buckthorn this year. So,
you did.
yes,
So
amazing.
it's a pain in the butt to walk through.
Yes, it is.
Interesting.
And my arms show it now.
Right. I liked that that you were growing the mycelium, or you're myceliating the chips, so that it felt like the fungus that you were kind of wrapping on the saplings already had a chance, right? So it didn't have to be like, well, I hope this, drop of spawn or the spores or whatever makes it. It's sort of like you gave it a head start by growing it on a chip. So it just had to sort of like jump from that to the other one. But I suppose you had to sterilize the chips first so that you could grow the specific species on it, right? And then attach them to the saplings.
Exactly. Exactly. And actually, we found that in some other studies that I didn't choose to highlight tonight, we did wood chips, and then we also did a myceliated grain on the opposite side of the stem at a different height. And we found that for a couple of these studies, the cankers that resulted around the grain inoculum were significantly larger than those that resulted around the wood chip inoculum. And so it appears that inoculum formulation is really important for either supporting the fungus or not giving it enough nutrient base to allow it to colonize.
Yeah, making it wants to jump to the tree if it doesn't have enough food in the inoculum, right? Very interesting. I was thinking when you're talking about sort of like inoculating a tree, wondering if you ever did, not like a cocktail, but more than one species. But I suppose once you've sort of weakened the tree, the spores and the mushrooms that are already present in the woods kind of do their thing once there's available dead wood.
Absolutely. Yeah, it's a whole fungal community in these cankers. And when we re-isolate, so I didn't share, but I didn't share this in the presentation, but for the greenhouse study, for example, we not only measured the size of the cankers for each fungal treatment, but we also have to re-isolate the fungus out of that canker to show that, hey, this fungus is here and it's causing this effect. And man, we get a lot of fungi coming out of those cankers, not just the one that we put in.
Interesting. So you have to prove that it's the exact one that you inoculated it for, but then you found all sorts of others.
Yes. Yep. Exactly. Yep.
Interesting. When I saw the hydro hatchet, I was like, man, I do a lot of shiitake logs. I'm like, I wonder if that's a quicker way, if you could market that tool to just use it on a bunch of
Oh, heck yeah.
growing oysters and other stuff on logs.
That's a great idea. Yeah.
Awesome. All right. Oh yes. CJ says that. Can't wait to hear more. I like the hydro hatchet. Yeah. Awesome. Do they make it in a toddler size yet? Awesome. Well, thank you again, Ryan. It's so fun to hear